if you see me typing, I'm going to be taking notes.
That's fine. All right. Well, hello everyone here in person, and hello everyone on the internet. My name is Matt Tatz, and today I'm going to be talking to you about mercury, salmonella, and urbanization, and how that relates to the American white ibis. Yeah, in a moment. Hold on, gotta get my clicker working. It's a Zoom kit. Okay. Everyone on Zoom, does it still look good? Presenter mode?
Yes.
Okay, awesome.
Yes.
Thank you very much. Okay, so an outline for this presentation, split into two parts. The first part is going to be between mercury and urbanization. So sources of mercury, where is it coming from? How is it ending up in the ibis, and how that relates to urbanization in the greater Everglades ecosystem. And then the second part is going to be between mercury and salmonella in the white ibis, and how those two factors might be related. So first, we're going to talk about mercury and urbanization. So most anthropogenic sources of mercury are emitted from coal-burning power plants, where they're released into the atmosphere. Here in the atmosphere, mercury can travel up to 100 kilometers away from the source before photochemically oxidizing into mercury oxide, this mercury too here. Then eventually that mercury oxide is going to end up in cloud formation before eventually wet depositing into ecosystems, such as natural wetlands, but also urban water bodies. Now, specifically, how they end up in urban water bodies, another source could be vehicular fuel emissions, which in Florida are not regulated. Essentially, because of all the impervious surfaces in urban areas, we could see a runoff of that inorganic mercury into nearby water bodies. And some studies have found that mercury can be converted to the organic form methylmercury within the cell. So now to talk about how that methylmercury organic form ends up in organisms and wildlife, the system of bioaccumulation. So we have that wet deposition into aquatic sediments, and it's primarily going to be sulfate-reducing bacteria that converts mercury into the organic form methylmercury. Because methylmercury is highly lipophilic, we see this transition in wildlife. In the ibis, that's going to be primarily aquatic invertebrates, like crawfish, their primary food source, but also small fish, which they forage on. In addition to sulfate-reducing bacteria, some studies have shown iron-reducing bacteria can also convert mercury to methylmercury, but that's not as frequent. And then we're going to see that increase in mercury concentrations as we go up in the trophic chain, eventually leading to high trophic species like carnivorous birds. Okay, so an overview of mercury in the greater Everglades ecosystem. Mercury has a high potential to get converted into methylmercury in wetlands because they go through this cycle of wet and dry seasons. And on the left, we see the Everglades habitat during the wet season, and on the right, we can see the habitat when it's a dry season. And essentially, this creates anoxic conditions during the dry season, and that's when we're going to see a lot of methylation of mercury into methylmercury, particularly in low surface water levels like the Everglades ecosystem. Now, in Florida, during the early 1990s, there was a period where they decided to do a lot of poor waste incineration practices. And this caused a lot of mercury to end up entering the atmosphere, where still to today, we see wet deposition of mercury into Florida ecosystems. There was so much mercury during this period that in 2002, there was a study done looking at water bodies in Florida, so rivers and lakes, and they found that 74% of these water bodies surveyed for mercury had a don't eat fish advisory issued to them, which is a large percentage. And so this caused a lot of public concern for human health and also for wildlife health. But because around the early 2000s, it was managed, these waste incineration practices, we've seen a reduce in the amount of advisories to not eat fish. And so research has calmed down a little bit, but it was very popular during this time. Okay, so when I'm referring to the greater Florida Everglades ecosystem, I'm referring to This line here in red, this is about most of Central and South Florida, and it encompasses that whole region. But in yellow, you'll see that the Florida state protected land waters only make up about half of that greater Everglades ecosystem. So there's a large portion of this land that is not protected. Now, in wetlands, we have peat, and peat is a very rich soil type, which is very good for agriculture. So we saw an increase in the amount of agriculture used of the Everglades habitat. And then the Everglades agricultural area was created, shown here in white. Also, because we have Lake Okeechobee, which kind of adds to the water levels of the Everglades ecosystem, this has been used largely in canals for irrigating these farms. So on this map to the right, we see the Everglades agricultural ecosystem here, right below Lake Okeechobee. And we have our system of canals. Right below the EAA, we see our water conservation areas, which are protected. But just east of those, we have urban areas like Palm Beach County, where increased urbanization has caused a further decrease in the ecosystem habitat quality. So our study species is the American white ibis, Eudocimus albus. It is a wading bird, and the population that we've studied in the Hernandez lab are primarily ibis native to Florida, where they complete their annual cycle on this continent. Though we do have some ibis populations in the Caribbean and also in Central America, the population we're currently studying are the ones circled here in Florida. Okay, so how has the white ibis become more urbanized? Naturally, the American white ibis is a nomadic forager. So they will seek out areas of high prey abundance where they primarily forage on crawfish, but they'll also forage on other aquatic invertebrates. Now, as the greater Everglades habitat, has decreased, we see basically the ibis rely on moderate and medium level of water height to forage for these crawfish. So when water levels are too low, we see a decreased prey abundance, and this has led some ibis population to forage in urban habitats, primarily during the non-breeding season. So here pictured is Disney World, where people feed these ibis things like bread. When I was in Florida, I saw people throwing out whole loaves of bread. I had one lady walk up to me and say her neighbors like to feed them raw meat that they don't use anymore. So they eat all sorts of handouts. They also forage in golf courses and in neighborhoods on terrestrial invertebrates and also on trash and landfills. So these urban ibis populations have a higher site fidelity than those found in the more natural Everglades ecosystem. But again, the ibis populations in urban areas are primarily foraging in these urban habitats during the non-breeding season. And then during the breeding season, they will go back to natural areas to forage. But this doesn't necessarily apply to urban populations that are either now breeding in urban areas, which we've seen recently, or if prey abundance is too low in the natural areas, they might decide to stay in the urban areas for part of the breeding season. Okay, so in the Hernandez lab, we have studied ibis a lot, and with collaborators. We've studied physiology and health, behavior, movement patterns and habitat use, productivity, and diet. Now, there has been research done on contaminants in the ibis, a lot of mercury work over in Florida, specifically, primarily with Dr. Peter Frederick. But we have not looked at contaminants in these ibis utilizing urban areas. So that's the focus of this research. Now, Frederick basically found that because the ibis is a high trophic species aquatic bird, that it well represents mercury contamination in sort of that higher level trophic area. And so he uses mercury as a way to indicate potential exposure. And we found because there's mercury affecting these birds, primarily reproduction in the natural habitats, that it would be appropriate to see if mercury is affecting these urban populations as well in these urban water bodies. Okay, so when people think about contaminant studies, I think that generally they're thinking about acute toxicity, which is going to be short-term exposure to a contaminant and generally a high concentration. And we do see, there has been studies on birds in general, looking at acute toxicity of mercury. Mercury, specifically methylmercury, since it's the organic form, but generally throughout this presentation, I'm just going to refer to this as mercury. But it's a neurotoxin that can cross the blood-brain barrier, so this causes a variety of neurotoxic effects, primarily decreased food consumption, poor muscular coordination, and spinal cord degeneration. Now that's fine and good. When we're looking at wildlife, generally we're more concerned with the chronic sublethal effects of mercury. Chronic referring to exposure over a long period of time, and sublethal effects referring to very low doses below the lethal concentration. So in Ibis, there has been a lot of studies done on the chronic exposure, these chronic sublethal exposure to Ibis. One study, and most of these are dosing studies, but one study found as much as a 35% decrease in fledgling production in these nests. And also, there's another dosing study that found male ibis, it was 55% of male ibis with high mercury concentrations formed same-sex pairs, and this created a 30% decrease in egg production. Also, interestingly, those ibis that had high mercury concentrations, whether forming same-sex pairing or not, there's actually less female ibis that selected for them to create a nest with. So that's interesting. Also, we have seen high mercury concentration cause altered foraging activities, though between control and high dosage groups, it wasn't really clear, but there was altered foraging activities happening. And then endocrine disruption in corticosterone, there we go, practice that one. Basically, interestingly, we found urban ibis had lower amounts of corticosterone, meaning that they were less stressed, and then the male ibis in these dosing studies had higher estradiol, which kind of makes sense, it goes hand in hand. All right. Now, there's been a lot of ibis studies that look at how mercury accumulates in the ibis in particular, and so I sort of want to talk to you about how we've seen mercury accumulate in the ibis, differences between sex and life stage, and then how this has impacted some methodology that researchers have used. So male and female ibis are exposed to mercury, but male ibis tend to be exposed to higher concentrations of mercury. Primarily, both sexes get rid of methylmercury by excreting it into the feathers. That's their primary pathway. But female ibis are also able to dump mercury into their eggs, and that's one way that they're able to offload it. Also, female ibis are smaller than male ibis, so they're generally consuming less prey with high mercury concentrations, and so that's another reason we would expect female ibis to have lower mercury concentrations. Because feathers is the primary excretion pathway, a lot of ibis research have used feathers to observe mercury concentrations in these populations. And then based on life stage, because mercury bioaccumulates over a time, assuming that the mercury accumulation is at a faster rate than their demethylation rate, we would expect to see that mercury concentration is going to increase as the individuals get older. So in this study, we're looking at adult ibis because adults are going to potentially have the highest mercury concentration and thus represent impacts of mercury on these populations. Okay, so there was a environmental risk assessment that was done on the Greater Florida Everglades ecosystem, looking at methylmercury concentrations, and they found that mercury was unevenly distributed across the Everglades. Basically, they found that mercury can be found in what they call hotspots. So essentially, certain areas of the Everglades ecosystem have concentrated mercury, while others are going to have small amounts. And because the ibis is a nomadic species naturally, this means that they're going to have sporadic exposure to mercury throughout their lifetime, because they might go to one site and forage where the prey has high mercury concentrations, but then they might forage on prey with very low mercury concentrations. Now in urban ibis, currently it's unknown, the amount of site exposure to mercury and also the amount of mercury in these individuals. However, we do know that these urban individuals tend to have higher site fidelity. So if these sites happen to have a lot of bioavailable mercury, that means we would anticipate that the urban ibis could have higher mercury concentrations than those found in the natural habitats. So we hypothesized that as urbanization increases at the capture site, that mercury concentration would be higher as well. Okay, so now I'm going to move into our methodology, starting with our capture sites. We had 15 capture sites, and these samples were collected between 2015 and 2017 as part of the NSF project, where we were sampling adult ibis. We sampled during the pre-breeding, breeding, and post-breeding seasons. And we were sampling in Palm Beach County, outlined here in red, Southeast Florida. And that's because, one, it encompasses the Everglades agricultural area, and we also know that there's urban ibis populations that are known to forage in this county. Also, Palm Beach County is the third most populous county in Florida, which is further increasing. So this well represents urbanization. So using GIS, I created a map, represent our capture sites. Here outlined in white is Palm Beach County. And then the sort of white overlay represents the Everglades agricultural area, which you can see takes up around half of the entire county. So that's quite a lot of land. And then the blue dots represent our lower urbanization sites. And then our red dots represent our higher urbanization sites. So you can think of low urbanization as more natural areas or natural habitats, and high urbanization as more urban habitats. Okay, so there had already been done some studies that utilizes urbanization, and an urbanization gradient was already created using land cover analysis by Kidweaver et al. So using GIS in the 2016 cooperative land cover map, land classes were divided into 3 categories: wetlands in blue, urban areas in red, and then other in gray here. And then using first passage analysis, we determined a buffer area of 2 square kilometers around each capture site to determine the level of urbanization. So down here at the bottom right, we can see a very low urbanization habitat. And then at the top right, a very high urbanization habitat. You can see a lot of residential area. So that's how our urbanization gradient was determined. Now, to represent this in a different way, here is our urbanization gradient from 0 to 68% urbanization. Our total number of individuals we sampled for this project was 94, but we divided this further into subgroups. So first, between 0 and 34% urbanization, we have our low urbanization subgroup with a sample size of 44. And then from 34 to 68% urbanization, we have our high urbanization group with a sample size of 50. Then to account for salmonella shedding or not shedding, we further divided these groups into 4 subgroups. Salmonella shedding or positive, and not shedding or negative here. And then the same for our high urbanization. And each of these subgroups have a sample size of 25, except for the shedding in the low urbanization subgroup. And that's simply because we didn't have 25 individuals who happened to be shedding in that subgroup. So this is how we captured urban ibis using a leg lasso technique. So these were baited using handouts such as bread, and then the ibis would step onto the lasso and be pulled in for capture. And I can replay that. This is kind of fast. Here again, there's a leg lasso out here. Once an ibis steps in it, we capture. Now this worked for urban populations because they were, more comfortable being around people and they were willing to get that close. But in natural populations, it was more difficult to capture ibis. One, because there's a lot more space to cover, and where these populations are is harder to predict since they don't have a high site fidelity like the urbanized ibis. So we use decoys to bring in these ibis. So here are lawn flamingos painted white, which we keyed the term snow mingos. And then we would have a modified mist net set up, either just a singular mist net or in a triangular or square pattern. These green arrows, I know it's a little hard to see, but the green arrows show the 2 ends of the mist net, which is set across here. And so the idea is that a natural ibis would see white dots below them and be like, oh, maybe these are ibis foraging. So they would come down and then they'd get caught in the mist net, and we could sample them that way. So we collected biological samples while we captured the ibis. So this includes fecal samples, where we either collected it when they would defecate on the ground or in the researcher's lab using a sterile swab. And then we were able to isolate for salmonella. Here's a positive and negative control. The positive control, we see these dark black colonies, that's representing salmonella. And then we also collected a secondary flight feather from these adult ibis during capture. And shown here, we stored those feathers in envelopes at room temperature, which I then used to analyze. And I have these stickers here representing our subgroups, orange being the high urbanization habitats, blue being low urbanization, and then I labeled them accordingly. So we used feathers to determine mercury concentration in ibis. One, because a lot of prior researchers used feathers, so it seemed like a reasonable measurement to take. But for other reasons, one, feathers represent up to 90% of the body burden of an individual at the time in which the feather was grown, since it's the primary excretion pathway. And this is a relatively non-invasive procedure. Also, ibis molt their feathers primarily during the breeding season, so once a year. They can molt up to, well, greater than 5% of their total body feathers at this point in time. Now, a limitation of using feathers is that it only represents the mercury concentration at the time in which the feather was grown. But for urban ibis, since they have high site fidelity, we believe that because they would primarily be spending a lot of their time at these sites, that this would well represent the mercury concentration at that time. So here's how I process the feathers. I sanitize the tools using a bleach dilution solution. Then I cut the feathers into tiny pieces as shown here. Essentially, we're homogenizing the sample to make sure that all parts of the feather are equally represented when we're doing our mercury analysis. Then we put our samples into a bead beater. So you can see here in the tubes are small feather pieces cut up very small. And this would shake vigorously to homogenize the sample. And then finally, once homogenized, these feathers would be put into a direct mercury analyzer 80, which is essentially a spectrophotometer. And it's a standard way to measure total mercury in samples. So this is a breakdown of how the spectrophotometer works. We put our initial sample into a boat here where it's dried and decomposed. Then it's moved into a catalyst furnace where sulfurs and nitrates are removed. Then it's put into this amalgamator where essentially the sample is flash incinerated. And then mercury is captured and brought up to this mercury trap where basically using the spectrophotometer, the absorbance of the mercury is determined. For these samples, that was 253.7 nanometers. And then to use as controls and to calibrate the machine, we had some standard reference materials, Torque 3, which calibrates the machine for methylmercury, and that's lobster hepatopancreas, and Pax 3, which is a marine sediment, and that calibrates the machine for total mercury. And again, because this machine found total mercury concentration, but for the purposes of this, I'm just referring to this as mercury going forward. Now, while we're not directly looking at methylmercury, but because methylmercury is the primary organic form that we would expect to see, I think it's around up to 90% of mercury found in an individual is generally going to be methylmercury, so that's sort of the comparison we're making here. Okay, so now I'm going to go ahead and move into our results. This represents the years in which the samples we analyzed were conducted. So we have year on the x-axis and the average mercury concentration. This is in milligrams or kilogram, but that's parts per million on the y-axis. And what we see here is between 2015 to 2017, there was no significant differences in the average amount of mercury between samples. So we don't think there was any influence of year on this. Now, this represents all three of those years as well, which was really, I know it spans 2015 to 2017, but it was actually just a 2-year sample size. We had 2 field seasons in the spring, 2 in the summer. and 2 in the fall. And again, we have average mercury concentration on the y-axis. And we see that there's no significant difference between seasonality for mercury concentrations in our individuals. Now, summer appears to be a bit lower, but I think that's just because the number of ibis we sampled in the summer was lower. And if we had a bigger sample size, it would probably even out. And then we also analyzed mercury concentration for sex. There was no significant difference between sex. We used PCR to determine sex of the birds. Average mercury concentration on the y-axis. And this was surprising to find that there was no significant difference because, like I previously discussed, we would have anticipated that female ibis have lower mercury concentrations because they can, get rid of some of that mercury in the eggs. However, there was no difference, and that's not necessarily a bad thing for this project, at least, because sex isn't, skewing our data for looking at urbanization. Okay, so here we have 2 figures. A represents low urbanization sites and B represents high urbanization sites. Now we have the average mercury concentration on the x-axis and then the percent of capture sites on the y-axis, which was 7 sites for low urbanization and 8 sites for high urbanization. And we see that the total mercury we found ranged from 1 to 4 parts per million in our samples on average. Now, what's notable is on our low urbanization sites, we see a wider range, a more uniform distribution of mercury concentration. So this spans from 1 to 4, and there was a low number of sites that had these different mercury measurements, meaning it was more variable. Whereas in our high urbanization sites, we see a significant number of sites had mercury concentration between 1 and 1.5. And this is a lot less uniformly distributed. So generally we're seeing a lot lower mercury concentration in these urban sites, well, in the high urbanization sites compared to the low urbanization sites. This is a linear regression. We have the average site urbanization score on the x-axis and the average mercury concentration on the y-axis. Our data points represent the average mercury concentration at each of the capture sites. So there's 15 data points represented here. And we see a significant moderate negative correlation between urbanization score at the capture site and mercury concentration. And then between our 2 subgroups of low urbanization and high urbanization, just to see if there's a technical statistical difference between these 2 subgroups, we have our low urbanization sites on the left, high on the right, and then on the y-axis, average mercury concentration. And we see that there was a significant difference between low and high urbanization sites. So if you recall, we predicted that as site urbanization level increased, that we would see higher mercury concentrations. But in fact, we actually saw quite the opposite. The actual relationship is as site urbanization level increases, mercury concentration decreased. Okay, so why might that be? We're going to go ahead and move into our first part of the discussion. So Oram et al., which is very useful for this interpretation, looked at sulfate concentrations surrounding the Everglades agricultural area. And here, the larger the blue circle represents higher sulfate concentration. Now, if you recall, sulfate reducing bacteria are needed to convert mercury to methylmercury, and this is creating a higher level of bioavailable mercury at these sites. And what we see is there's a lot more sulfate available just south of the Everglades agricultural area in canals. And this becomes less so as we move more into urban areas like West Palm Beach, this area near the coast. So I've included our capture sites here. In green are our low urbanization capture sites, and then in red are our high urbanization capture sites. And what we see is that our low urbanization sites are a lot closer to the EAA than our high urbanization sites. And this makes sense because this is surrounding the conservation areas, and of course the urban areas are along the coast. Now there is one exception, Lion Country Safari here. This is this red point over here, but I looked at that for mercury concentration individually to see if it would be higher, and it was not. Now, the premise is that since the high urbanization sites are farther away from the sulfate concentrations, that there may be a lower bioavailability of mercury in those sites, which would make sense on why we're seeing higher mercury concentrations in the ibis that are primarily utilizing the low urbanization habitat. Another reason could be the whole dry versus wet season. So in these natural wetlands, we see that annual cycle of dry and wet seasons, which creates those anoxic conditions, which are going to help methylate any mercury there. But in these urban water bodies, we don't see this same cycle, especially in things such as retention ponds, which are only drained like once a decade. And so those anoxic conditions aren't going to be as present as in the low urbanization wetland sites. And then also considering what are the urban ibis eating. So they're supplementing their diet with handouts, and human food is regulated for mercury. So if they're eating more handouts, we can expect that they are getting less exposure to mercury. And then also the terrestrial invertebrates that they may be feeding on in the low urbanization sites, since there's less bioavailable mercury, their prey likely has lower mercury concentrations as well. So it would make sense that the ibis found in the high urbanization sites have lower mercury. So I propose that there's a lower bioavailability of mercury in capture sites found in high urbanization habitats. All right, now looking at other research, Frederick et al., he sampled wild ibis in the greater Florida Everglades ecosystem prior to the waste incineration practices and after. So the first period was before 1990 and the second period was between 1998 and 2001. He found that before the practices, the waste incineration, there was a mean mercury concentration of around 1 part per million. And then after the incineration practices, which would have been the late 1990s, early 2000s, a much higher average concentration of 7.5. Now in our study, which was conducted a bit more than 15 years later, after the waste incineration practices had been managed, we see in our low urbanization sites an average concentration of 2.5, around 1.5 in our high urbanization sites, and an overall mercury concentration of around 2. And so we see that this average is much closer to the prior to the poor waste incineration practices, meaning that the management was likely effective, that just 15 years later, we saw a near decrease of 5.5 parts per million of mercury in the ibis, which is great. It's very significant. Now, there was another free-living ibis study conducted in 2005, where they were just looking at mercury concentrations in the greater Florida Everglades ecosystem, and they found a range of exposure between 0.3 to 20 parts per million, and they found in those ibis with higher mercury concentrations, higher nest abandonment. Now, this is not the average mercury concentration like I've been referring to. This is representing the range between individuals. So we saw a range in low urbanization sites between 0.25 and 8.5, and in high urbanization sites from 0.2 to around 7 parts per million. So on the higher end of our range, it seems like this is a lot higher, but I still think that those ibis with the highest mercury concentrations could potentially still see negative reproductive effects, like nest abandonment. And another study done by Frederick and Giacenna, it was a dosing study. They found that in their low dosage group, which was between 4 and 7 parts per million of mercury, that there was less eggs being laid, which is pretty comparable to the higher end of our individuals exposed. So it's also possible reproduction could be negatively affected in those individuals as well. Okay, so this figure, we have our capture site urbanization score on the x-axis and mercury concentration on the y-axis. And we have so many points because this is actually representing our individual mercury concentrations in our birds. So not looking at averages currently, just individuals. What I want to point out is that we had 5 individuals in our high urbanization range that had much higher mercury concentrations than the majority of individuals found in that category. And what this likely did was raise the average mercury concentration of our high urbanization category. The reason I think that these individuals likely had a high mercury concentration is, if you recall, generally urban ibis are going to be foraging during the non-breeding season, but it's possible that these ibis were foraging in natural habitats for a while. It's likely they were foraging near those hot spots, and then they just were sampled in the high urbanization sites, which is why we see such high concentrations. Because if you look at our low urbanization sites, you know, it's much more comparable here. So I think that's likely what happened. Okay, so the second part of our presentation is going to be moving away from urbanization and mercury and more towards salmonella and salmonella dynamics in birds. Okay, so generally when we're looking at wildlife health, in ecotoxicology, we're going to be looking at the impacts of contaminants. And in disease ecology, a lot of times we are looking at the impact of pathogens on wildlife health. But not often, at least in birds, do we see a study on the interaction between contaminants and pathogens in individuals. And that's important because in wildlife, like in the ibis, we're going to see simultaneous exposure to both of these factors. Previously, we have studied, well, Frederick et al. Had studied mercury in the natural ibis populations in the low urbanization. And we have studied salmonella in the ibis populations, but this study seeks to look at the possible interaction between mercury and salmonella. So does mercury decrease immune function? The answer is likely yes, but not very convincingly so. The research that is done has had little replication, so generally there's just one or two studies doing a particular immune system test. Some of these results tend to be conflicting, and no current mechanism is determined. So some of these studies hint that mercury may cause immunosuppression in birds, but it's not very conclusive at this time. Now, of those tests, I first want to point out that there's no one test that can look at the overall impact of mercury on the immune system. Generally, we're looking at individual tests that are looking at one particular part of the immune system. Now, we've seen impacts to both innate and acquired immunity in birds. So this could be for phagocytes like macrophages and heterophils, where phagocytosis was impacted, or in lymphocytes like B cells and T cells, where proliferation was impacted. But again, when these studies are done and they're looking at one particular part of the immune system, it can be hard to interpret the actual effects of mercury on the overall immune system. One aspect that mercury may impact immunity in the ibis could be through the gastrointestinal microbiota. That's a mouthful. So its communities are made up of primarily bacteria, but also protists, yeast, viruses, and archaea. And they have a symbiotic relationship with their host because they can provide nutrition and defense. Now, they provide defense primarily through competitive exclusion. By having a healthy gastrointestinal microbiome, we can exclude pathogens that are coming in directly, or indirectly because they can essentially alert the host if there's incoming pathogens, which can turn on the immune system in a sense. Now, there was a study looking at the gastrointestinal microbiome of ibis, a free-living ibis, and we found that natural ibis had a more diverse gut microbiome than urban ibis, which had a less diverse gastrointestinal microbiome. So essentially that means that if there's a less diverse microbiome, we may see part of that could be because of mercury. Now that's further supported by recent studies on rodents and fish, so lab rats and mice, and this one fish species here, where these were dosing studies, and they found that these species that had higher mercury, these individuals that had higher mercury concentrations had a less diverse gastrointestinal microbiome. Then there was another study done on yellowfin sea bream, and they found that a healthy gastrointestinal microbiome increased demethylation rates of mercury. So I'm getting the idea that as diversity increases in the gastrointestinal microbiome, this is going to help offset the toxic impacts of mercury on an individual. So basically, if we have a less diverse microbiome due to mercury, and we potentially see impacts on immune function because of mercury, This could potentially increase pathogen infection and shedding when mercury concentrations are high. Now, the pathogen that we're specifically looking at is Salmonella. It's a gram-negative bacillium motile bacteria. It has greater than 2,500 serotypes, and we have studied Salmonella in the Ibis previously. Salmonella is important because it can cause negative health effects in humans, and it can cause mortality in aquatic bird nestlings. Okay, so previously in our lab, we looked at the impacts of Salmonella essentially between low and high urbanization sites. This graph, this figure is showing on the x-axis the proportion of opened or developed land cover, which is essentially saying more urbanized habitat type. And we have Salmonella prevalence on the y-axis. And we found a significant positive correlation that as the amount of open land cover increased, that there'd be a higher Salmonella prevalence, open developed land cover. And recently we've determined that Ibis have a mean prevalence of around 26%. So for our second hypothesis, we hypothesized that Ibis that are shedding Salmonella would have a higher, potentially have a higher mercury concentration. Now, in the first part of the presentation, we found that urbanization has an effect on mercury concentration. So I wanted to make sure I had control for that. We're going to go ahead and move into our results now. Salmonella shedding and status had already been determined previously. So I'm just going to go ahead and show how mercury and Salmonella status related. So in these graphs, I have low urbanization, our high urbanization site, and then overall urbanization. We have shedding status on the x-axis, negative and positive, and then the overall mercury concentration on the y-axis in these box plots. And across all of the urbanization groups, we found no significant differences in mercury concentration based on Salmonella shedding. Now going to move into the discussion on why we might have seen that. First, it's possible that the type of immunosuppression caused by mercury simply has no impact on Salmonella dynamics in the white ibis. That could simply be the case. However, I think there were some potential experimental design impacts that could have caused this result. We were studying wild ibis, and so by doing so, we are sampling active individuals in the population. Previously, impacts of high mercury showed altered foraging activity. So it's possible that the ibis we were currently sampling were skewed, essentially. And then this is also important. We were specifically analyzing the impact of mercury on Salmonella shedding, so shedding or not shedding, which is very specific. So we did not analyze the impact of mercury on the gut microbiome. So this doesn't mean that mercury has no impact on gut microbiota, or sorry, gastrointestinal microbiota in birds. That could still very likely be happening. Now, most ibis studies that found significant impacts of mercury, primarily on reproduction, were dosing studies. Now, the problem with dosing studies is they remove environmental challenges because you're taking a captive population, and so there's a lot of additional factors that you're not accounting for. So there's some drawbacks to both styles, but we have seen more effects when doing dosing studies on the ibis from mercury. Okay, so in conclusion, we found that there was a negative correlation between mercury concentration and site urbanization level. We concluded that urban ibis captured in high urbanization sites were exposed to mercury, even if it was in lower amounts than in the low urbanization sites. And we proposed that there's less mercury bioavailability in low urbanization sites, at least in this area of the greater Florida Everglades ecosystem, as it's directly related to its, or potentially directly related to its distance from the Everglades agricultural area. We found there was no difference in mercury concentration between ibis that were shedding and not shedding Salmonella, but we proposed a controlled dosing experiment that can essentially find, is there an impact of mercury on the gut microbiome? And I have the slide prepared during the Q&A if you'd like to discuss that further. Now, moving on to future research and benefits. This picture just shows how messy fieldwork can get. This was funny, so I'm about to show you that I did soil core sampling. There was one site that I essentially sinked about a foot down into this soil, and there was one path that was leading to the area that I needed to sample based on the coordinate point. And it turns out I could have just gone around, and I would have never dealt with this. But that was about 30 minutes of very wet walking. And then this is just a white ibis on its nest in an urban mercury. So I took soil core samples. I took soil core samples nearest the road, the nearest water body, and in between these 2 points at each of our capture sites. And the reason I did this is because we wanted to see if the mercury concentrations we found at the capture sites would be directly related to the mercury concentrations found in individuals at those capture sites. But currently we have not yet analyzed these samples, but it's something we may consider doing going forward. And then the benefits of this research, it's possible that mercury can impact the health and specifically the immune system of the ibis. We know that ibis in high urbanization sites have the potential to transmit salmonella to conspecifics and also to people. And because people currently seem to have a good relationship with the ibis, at least in Palm Beach County, that presents a possible health concern to people as well. But also we want to talk about the cohabituation of urban populations and people as well, especially as the Everglades ecosystem quality continues to dwindle. It's possible we'll see more species becoming more urbanized over time. So I think it's important to determine how people feel about these populations and, looking at perceptions of risk and things along those sorts. And so we're looking at mercury because mercury may impact salmonella dynamics and that all these factors go together. We just want to make sure we can sort of cross off all the bases. And then the Florida Fish and Wildlife Conservation Commission proposed that we continue monitoring of mercury in wildlife, and they specifically noted a need to look at aquatic bird nestlings mercury concentration as well. So I want to say thank you to first the Georgia Ornithological Society for funding my research, which is excellent. The Savannah River Ecology Lab and Stacy for providing costs for the feather analyses. NSF for providing, sort of helping build this entire study system and providing the cost to acquire the salmonella status of these individuals. My committee, Sonia, me, and Stacy for supporting me through this research. And as we go forward within it as well. Jason at SREL, who helped train me on homogenization of the feather samples, and Angela, who ran my samples through the DMA 80 for me. To the overall Hernandez lab for supporting me emotionally and in my writing and all sorts of other ways throughout this whole process. To Kate at the Odom School and Kim at the Franklin College, who has helped train me in my teaching and helped providing me with assistantships to fund my education here at UGA. And also to Carly at Cedar Schultz High for working with me on outreach of this project, where I have given multiple lectures to high school students in an AP environmental science class about urbanization and conservation. And with that, I thank you for your attention, and I'm happy to take any questions.
Good job, Matt. This is the time for general questions, either from folks here or on Zoom.
Should I stop sharing or should I? What do you think?
Yeah, probably stop sharing and just look at you.
Okay.
I do have 2 questions for you.
Okay.
Were you ever able to see if Maureen's microbiome work overlapped with these ibis?
I've not yet taken a look at that.
Was her microbiome work done for 2000? Okay, so you should be able to.
There should be some animals that do overlap, but yeah, some of them, like sometimes we get feathers, sometimes we get it, and feces. you know, so
Yeah.
you know.
So hopefully that
Actually, we
should
did
be-
that on cells, so actually we should have, you should see some overlap.
That's something I want to do early on into the PhD going forward, since we talked about
Well,
that.
it might be something actually for the paper,
Oh, really? Okay.
yeah, for the manuscript.
Then over the summer.
So you'll hopefully have some full preliminary data when you do that to answer.
Yeah, that would be
your
excellent.
question. And I found it interesting that you decided to do a graph on seasonality and mercury concentration, even though all the feathers are deposited generally around one season. Was there a reason you did that? Would you like
Well,
another sample?
yeah, so actually the reason we looked at seasonality was specifically because they're all deposited at one season. So, you know, I've looked at a lot of the research and there's really only one report of the amount of molting occurring during the breeding season.
Mhm.
And so essentially by looking at seasonality, I was hoping to see there would be no differences between them because say mercury concentration was a lot higher at one season, that essentially by finding no differences between them, it supports that they are all occurring at the same time, which is what we wanted to see. Does that make sense?
Yeah.
Okay.
Those are my two.
I have a question. I was wondering if you have like an idea of how different the gut microbiome of IBCs may be, whether they're urban or non-urban, and whether that relates.
Yeah, absolutely. So there was that one prior study done on free-living ibis where they actually looked at the gastrointestinal microbiome in urban versus non-urban birds, and they found that the diversity of the gastrointestinal microbiome was lower in higher urbanization sites. And this is likely attributed to, you know, handouts where it's going to cause a less diverse exposure. For sure. So I would anticipate.
And the other thing that was important about that is that the community composition of the bacteria would impact the diversity, which actually was negatively correlated with salmonella problems.
Yeah.
So that was one of the impetus, the impetus for looking at this, because we had a direct link between the gut microbiome and salmonella already.
Yeah.
Thank you for the question. Happy to take questions on Zoom as well, if anyone has any. And yeah, I've got the chat open.
I know some other groups, and correct me if I'm wrong, like we have a collaborator in Spain and he keeps eggs that never hatch to do toxin testing on. Would that be something you could easily sample at urban and natural colonies to look for like
Mercury within the eggs themselves? I mean, we would likely have access to these eggs. It depends on if we'd be able to take them with our permitting. I would imagine we would we could do both habitat types, but from what I understand, there's a lot more reproduction in the natural habitats in the low urbanization. So if we were concerned we might impact reproduction by taking too many eggs, say in an urban rookery, maybe we'd start in the low urbanization sites. But I do think it would be worth looking at, because there was one study that basically looked at the sensitivity of mercury across all sorts of species of birds and eggs. And white ibis was considered highly sensitive. So through that.
In terms of being an indicator of the environment, like matching with environmental mercury, is that what you're meaning there?
Yeah, I'm not, it was a lab study, so I'm not sure. I didn't really look into the implications of it. From what I gather, they just were looking to see, I suppose, how, I don't want to say anything wrong, but I think it was how long the individual, you know, survived or any impacts to that egg based on mercury concentration.
Oh, like embryonic death.
Yeah, exactly.
Okay, sorry.
I'm pretty sure that's what it was related to.
Okay.
In which case the ibis, it took a lot lower concentration to cause that.
You may get with Julia and she can talk to Lindsay about the number of eggs that don't hatch and how easy it would be to get a hold of those.
Oh, true.
Like
And the urban habitats.
And you won't necessarily be affecting the reproduction because these are failed eggs.
Yeah.
They're not gonna hatch. They're like left in the nest and they
Oh,
can
right.
rot.
Right.
Like, the problem is transporting the eggs, so it's not disgusting by the time.
Yeah.
you get it. Yeah.
Mia has a question for me in the chat. She says, regarding my methodology, is there anything I would do differently? going forward. And yes, Mia, I think, and from what I've read, there's some discussion that looking at feathers in adult bird individuals is highly limited because it's, at least comparing it to the capture site, because it's, you know, the feathers are grown at the time in which the mercury is deposited at the time in which the feathers are grown. So there's potential, you know, confusion in terms of interpreting your results that way. So a lot of studies recommend, or at least one study recommended, looking at blood of these birds because it's giving you a more immediate look at mercury concentration at the time in which you're sampling. So if I would do it, again, I would probably look at blood, or I might have looked at blood in addition to feathers and seeing how those